Revision video for IB biology, and this is a HL only video. We'll be looking at cardiac cycle, the heart, tissue fluid. And remember, revision support to help you with your understanding. You need to do your own independent study in order to get good grades. So we're going to start off looking at tissue fluid today, and this picture quite nicely shows you what tissue fluid, how tissue fluid runs out back into the body through something called a lymphatic system. So if we start off then with tissue fluid, what is tissue fluid itself? When blood comes down from the arteries into the capillaries, which is where exchange happens, larger molecules cannot leave the blood, so blood cells and blood proteins go straight through and then that way. But smaller substances can freely diffuse between the tissues and the blood. So amino acids, oxygen, carbon dioxide and water can easily go in and out in between all the tissues as a result. Some fluids leak out of the blood into the tissue fluid as well, and water cannot be allowed to freely leave the blood, so it has to be controlled by water potential. So plasma is what the fluid is called around the blood cells. Okay, so this is what the plasma is here. Okay, and effectively when you look at your whole body's water, you have about 42 litres of water in your body. 14 litres of it are extracellular, okay, and that includes intestinal fluid, plasma, and transcellular fluid. And if we look at it and how it works, we're basically pushing out of the capillary. There is a high amount of hydrostatic pressure forcing the fluid out into the tissues, and pushing it back in, there is less hydrostatic pressure, so it's linked to that being decreased, and water potential draws liquids back in through with the water. So again, as we want to know, if you want to ask how is it likely to leave in the arterial end of the capillary, there is high hydrostatic pressure near the arteries, and that forces fluid tissue out into the tissue spaces. There is some opposition from water potential, but it's not great overall. So the green here is greater, forcing liquids out, than the water potential forcing it back in. However, as you go down away from the artery end of the capillary, the hydrostatic pressure massively decreases. So at this point, there is lower hydrostatic pressure as there is less fluid left in the capillary. So the hydrostatic pressure drops, so little is leaving, but you still have a water potential pulling water back into the capillary. And at this point in the capillary, the water potential will be very, very low. And the reason the water potential will be low here, so low water potential, is because we've lost a lot of the fluid into the tissue, into the spaces between the tissues, and therefore the blood has got loads of proteins in it, but very little liquid left in it at this point, so it draws back in via osmosis. Numbers to this. So if we look at the arterial end of the capillary here, you have this numbers here. Okay, so the hydrostatic pressure is 4.3 in the capillary end, in the arterial end of the capillary. It's 1.1 in the tissue area. So you've got a change there of 3.3. So you've got a change there of 3.2 leaving outwards, whereas your inwards one is minus 2. Okay, and then so overall 3.3 minus 2, 1.2 out, whereas at the venous end you have the decreased pressure of hydrostatic pressure, so there it's only going to be 0.5, but you have minus 2 going out, so overall minus 5, which means it's an inflow of 1.5. The lymphatic system then is something that is quite important. So as we're saying, some of the water re-enters via the venular end via osmosis, but not all of it. So excess tissue fluid drains into the lymphatic system, and this is a network of tubes that transports excess fluid back into the blood vessels at places where it's safe to do so, mainly in two ducts close to the heart in the vena cava. So the fluids that do not leave and go back to the blood go back via the lymphatic system, and here you can see the lymphatic system joins back up with the blood system in the areas near the vena cava, in the vena cava near the heart. So the key thing is then how is it moved again? So there's no pressure in there in terms of hydrostatic pressure from the heart, but there is hydrostatic pressure from the tissue fluid leaving the capillary. So the tissue fluid leaving the capillary is under high hydrostatic pressure that forces the fluid into the lymphatic system, and then as you walk, the contraction of your body muscles push the fluid along the lymphatic system, similar to the vein. Now, you also need to know about the structure of the heart and be able to talk your way through the structure of. If we start from the left-hand side of the heart, the deoxygenated blood coming from the body after doing respiration contains high carbon dioxide, low oxygen. That enters via the vena cava. You've got inferior from below the heart, superior from above the heart. That enters into the right atrium. The right atrium, then the blood flow goes through the atrioventricular valve into the right ventricle, and then when the ventricle contracts, it forces blood out through to the pulmonary artery, and that goes to the lungs. From the lungs, the blood comes back via the pulmonary vein, now oxygenated after the exchange at the alveoli of the lungs, and that goes into the left atrium, which then allows blood to go into the left ventricle. This is much more thicker. You can see here the muscular wall is much more thicker, giving a stronger force of contraction, making the blood leave going via the aorta to the body under higher pressures. And again, the valves work in a similar way. They are pocket valves, and the blood closes the valve if the pressure is high in the wrong way to stop backflow of blood. So the muscle in the heart is called cardiac muscle, and the wall of the atrial ventricles have branch cells and connections between plasma membranes, allowing electrical signals to propagate the cell wall. This is allowing coordinated contractions. This type of muscle is unique in the heart. It can contract without stimulation. The heart is myogenic, so it doesn't need to be stimulated from motor zero. So your brain, whilst it can control the heart and speed it up or slow it down, doesn't create the 70 beats per minute on average. You may be asked to do a dissection, and you should be able to do a scientific diagram of the heart, and here you can see the rules of a scientific diagram. They should take up the space given, be drawn in sharp, clear lines, continuous lines, demonstrate appropriate sizes of the object, be fully labelled, and the label lines must be drawn with a sharp pencil, ruler, and not crossover. Scientific diagrams do not contain any shading or colouring. So then the maybe more challenging bit for a lot of people is the cardiac cycle. So you need to know the stages in one heartbeat and what control of it to start off with. This is how does the electrical impulse actually cause the heart to beat. Well, the natural pacemaker of the heart is called the sinoatrial node, and it is found here in the top of the right atrium, and it's a bundle of nerve cells together called a ganglion. And effectively, the sinoatrial node stimulates a wave of electrical excitation which spreads across the muscles on these lines you see here of the atria, causing the atrial muscle to contract, forcing the blood from the atria down into the ventricles. This wave spreads along the muscle walls of the atria, but it can't go straight down into the ventricles because there is a layer of non-conducting tissue here between the atrial and the ventricle. So the wave of excitation travels through to this part here called the atrioventricular node, which is the only way down to the rest of the heart. This node delays the contraction going down, and that gives it time for the blood to fill up the ventricle. The wave of contraction then continues, and it travels down specialized tissues called the Purkinje tissues down the septum to the base of the heart, or the base of the septum, where contractions start moving from the bottom upwards, forcing blood up and out of the ventricles through either the pulmonary artery or the aorta. And that is the control. one's got a different name. So if we use this example here, we'll start off with the yellow box here, which represents this yellow box here. So at first the heart does something called diastole, which is when the heart is relaxed completely and the atria and ventricles are both relaxed. And here you can see as a result blood comes into the atria and some starts trickling down into the ventricles. We then enter atrial systole, which was the atria start to contract and the ventricles stay relaxed, causing a higher pressure in the atria. Remember I said higher there, which means I'm comparative in languaging, higher pressure in the atria compared to the ventricles, so the blood moves down into the ventricles as a result of the higher pressure being caused by the volume in the ventricles decreasing. We then enter ventricle systole, which is where the atria are now relaxed and the ventricles contract. The atrioventricular valves prevent backflow into the atria, and the higher pressure in the ventricles compared to the blood vessels where it's leaving, either the pulmonary artery or the aorta, means the blood leaves by the semilunar valves out of the heart. And notice when it's at rest we call it diastole, when it's contracting we call it systole. So we have double diastole, atrial systole, and then ventricle systole. To describe a graph like this and explain it fully. So we have three lines here. We have this red line which shows you the pressure in the atria. We have the orange line showing you pressures in the ventricle, and we have this red line showing you the pressure in the aorta, which is always a lot higher. And if we see here, we're in atrial systole to start off with. So this is when the atrial is contracting. You can tell that because the atrial line here is higher pressure than the ventricle line. So the blood is moving from the atrial to the ventricle. And at this point, the atrioventricular valves are open. Until this point here, where the ventricles start to contract, the volume goes down as well, and the ventricles increase in pressure. The pressure increases above that of the atria, so the atrioventricular valves have to close. The pressure then rises really high, but no blood at this minute is leaving the ventricle until it overtakes the pressure of the aorta here. And the pressure then, the semilunar valves open, allowing the blood to leave, which then eventually, once the blood runs out, starts to decrease the pressure of the ventricles. Once the pressure drops back below the aorta pressure, the semilunar valves have to close to prevent backflow, and then the pressure drops as the ventricle relaxes, and at this point the atria start to fill back up with blood, and at this point here the atrioventricular valves open because the pressure in the atria becomes higher than the pressure in the ventricle, and the cycle starts again. Now for this video I've had to say that very quickly, but you must learn this graph and be able to talk your way through. You'll also be able to calculate your cardiac output, which is your stroke volume, and that is the volume of blood pumped to each heartbeat times the heart rate or beat per minute. So effectively, you might have a question like this: calculate your cardiac volume if you have a stroke volume of 70 and 75 beats per minute. So you just do 70 times 75, which will equal this bit. You may also be asked to rearrange the question as shown in the examples below.